what's happening here is that GTP is binding and GDP is being released. So the GTP is replacing the GDP. So there isn't any sort of kinase activity going on here. It's a simple replacement of a molecule. And since GTP in general is at a higher concentration in the cell than GDP, this is a more likely reaction to occur. That answer your question? Yes?
What's the difference between a G protein and a GTPase?
So a GTPase is a generalized molecule that all it has is a GTPase activity. Okay. A G protein is a subset of GTPases that follows kind of having an inactive form and inactive form. Any other questions? All right, so then the second major type of receptor signaling pathway that I want to talk about are G protein coupled receptor signaling. And so G protein coupled receptors they, just like other receptors, have a ligand binding domain. They have a G protein binding domain on the inside of the cell. And what links the two of these domains are actually 7 transmembrane domain regions. So G protein linked receptors all have a very similar structure. And again, the idea is that the ligand binds to the ligand binding domain. That causes a conformational change that is transmitted through the 7 transmembrane domain region into the GTP binding domain. And that change in the GTP binding domain region is what allows the G protein to be active. So then here is a schematic of a G protein coupled receptor. Again, here you have your ligand binding domain, your G protein binding domain, and your 7 transmembrane domains. So again, the ligand binds to the ligand binding domain. That binding causes a conformational change, which gets transmitted through the 7 transmembrane domain region to the G protein binding. So what ends up happening is that once that happens, so again, here we have our G protein coupled receptor. Here's our G protein. This G protein, unlike RAS, is heterotrimeric. So there are 3 subunits, an alpha subunit that binds to the nucleotide, and the beta gamma subunit. And so what ends up happening is that when the ligand binds to the receptor and activates it, that causes that conformational change, which that gets transmitted to the G protein, causing it to release GDP and bind GTP. That causes the G protein to release the receptor and also split the alpha and the beta gamma complexes. And so now you've got these 2 molecules that are free to act away from the receptor. So again, the idea is that the G protein linked receptors, by definition, are linked to G proteins. Specifically, these are heterotrimeric G proteins. pair, meaning there are three of them, hetero, meaning that they're different. There's an alpha, a beta, and a gamma subunit. It is the alpha subunit is what binds and hydrolyzes GTP. And again, the important thing is that it's the activated receptor which converts the alpha, beta, gamma bound to GTP to the alpha GTP bound plus the beta gamma, now separate molecules. And so then what class of molecule is the activated receptor acting like? Well, what type of molecule helps a G protein release GDP and bind GTP? A GEF, right. So in essence, the activated receptor is a GEF. Works in the exact same way. And so just like the small G proteins that we saw, this gets turned off by hydrolysis. And there are a class of enzymes that help with that. They're called RGS proteins, for regulator of G protein signaling. And these are basically gaps. They're gaps that are specific for heterochimeric G proteins. And so again, just like we saw with the RAS protein, small G protein, this is a point of fine-tuning. Again, because the RGS can decrease the signal strength by activating the hydrolysis. Any questions about that? All right. So, again, an important thing about this is that what ends up happening is that once the receptor gets activated and activates a G protein, again, that G protein dissociates from the receptor, so activated G protein. Okay, so that means that activated receptor can bind to an inactive G protein. And so what that means then is that any one activated receptor can activate multiple G proteins, which means this is a point of what? Amplification, right. Any questions about that? All right, so then you've now got these activated G proteins. You've got the alpha subunit and the beta gamma subunit. And so both the alpha and the beta gamma subunits can go on and activate other proteins. And oftentimes these other proteins, these other parts of the pathway they activate are called second messengers. In essence, they are the second phase of the signal transduction pathway. So one of the more common second messengers is cyclic AMP. And while cyclic AMP itself has no catalytic activity, it can activate other proteins, specifically protein kinase A. So then how is it that cyclic AMP gets created by the active G protein? Well, cyclic AMP gets created by an enzyme called adenylyl cyclase. And again, what adenylyl cyclase does is it converts ATP into cyclic AMP, and it itself gets activated by the G protein. Any questions about that? Now then, because adenylyl cyclase, once it gets active, can convert multiple ATPs into cyclic AMP, adenylyl cyclase is a point of amplification. And all those cyclic AMPs, they again can go and activate protein kinase A. Also just called pKa. pKa, being a kinase, can phosphorylate multiple molecules also. Which means that this too is a point of what? Amplification. And because it is a kinase, and whenever it gets phosphorylated, it can be dephosphorylated, what does this also represent?
Fine-tuning.
Reset. And the final thing I want to say about this is that just like there's a molecule that creates cyclic AMP, adenylyl cyclase, there is a molecule that destroys cyclic AMP called phosphodiesterase. And since this is something that actively destroys the second messenger, this is a point of what? Reset. Because again, you're turning off the signal. Any questions about that? All right. So then, how does a cell actually use this? Well, you actually have kind of looked at this in the lab when we take a look at dictyostelium aggregation and development. So, take a look at the surface of a cell, surface of a dicty cell. On the surface, there is a receptor which binds cyclic AMP. What receptor is that? CAR, right? Basically CAR1, cyclic AMP receptor 1. It is bound to a G protein. Has an alpha subunit called alpha 2, a beta, and a gamma subunit. So normally, this is bound to GDP. However, when the cyclic AMP binds, it activates a G protein, splitting it. So alpha 2 subunit bound to GTP. And the beta gamma subunit. that beta gamma subunit goes and it activates adenylyl cyclase. And what does adenylyl cyclase do? It makes cyclic AMP, right. So it takes ATP and creates cyclic AMP. And actually what it does is that cyclic AMP gets secreted outside the cell to act as a chemoattractant, right, to bring other cells towards it. But in addition, that cyclic AMP is used internally. That cyclic AMP, as we just found out, can be used to increase protein kinase A activity. And that protein kinase A actually ends up going into the nucleus and activating transcription. So here is our nuclear membrane. Goes into the nucleus, and it can phosphorylate transcription factors. And now, since you've got these new transcription factors that are on, this can now transcribe genes that are required for development. Now, since this involves this is a relatively slow signaling process. So it takes a little bit of time for these developmental genes to get ramped up. Yes?
What does the beta gamma subunit do in response to folate?
Actually, so in response to folate, it works the same way, except for instead of the CAR1 receptor, it's a folate receptor. And instead of G-alpha-2, it's a different G protein called G-alpha-4. But the beta gamma, oh, you mean why? Does it actually have a job? So actually, no. So adenylyl cyclase only gets activated during starvation. So it's only activated in response to the AMP. So the beta gamma might be doing something else during folate chemotaxis, but as to what that is, we don't know. Any other questions? All right, so again, this is kind of just half the story. This is what allows for the cells to start making transcribing those genes required for development. But also, what do you know happens to a cell that senses cyclic AMP? What does it do? It moves towards cyclic AMP. So what that means is that somehow chemotaxis has to be activated. And that is done by the alpha-2 subunit. So the alpha-2 subunit actually, it activates an enzyme called guanylyl cyclase, converting it from the inactive form to the active form. And guanylyl cyclase, as the name suggests, makes cyclic GMP. So it takes GTP and converts it into cyclic GMP. Now then, that has two jobs. Actually, well, has one job. That main job is to activate the phosphorylation of myosin 2. And myosin 2 phosphorylation happens in the rear of the cell. And that eventually leads to actin contraction, which is required for motility. Right, that last step, the traction step at the back, the squeezing of the toothpaste, that is what happens there. Now then, again, in addition to having to have traction, remember the cell also has to send out pseudopods. And again, that is also done